What is cytology?
Cytology is the branch of biology concerned with the structure and function of plant and animal cell. Cell is the basic unit of life. Cell, tissue, organ, system, and organism. What are the important terms?
First is protoplasm, the colorless material comprising the living part of a cell, including nucleus, cytoplasm, and other organelle. Second is protoplast, plant cell without cell wall. Third is cytoplasm, material within the living cell, excluding the nucleus. And fourth is the nucleoplasm, the liquid and organelle in the nucleus. The next is cytosol, aqueous portions of cytoplasm contain water and dissolved substances. The next is organelle, specialized or organized structures of a living cell. 7. Prokaryotic cell, single cell organism with neither a distinct nucleus with a membrane nor other specialized structures. 8. Eukaryotic cell, organism consisting of a cell or cell with genetic material is DNA in the form of chromosome contained within distinct nucleus. What is cytoplasmic membrane and cell wall?
Thin structure that completely surround the cell. Typically 7.5 to 8.0 micrometer thick. Structure, fluid mosaic model. The functions are first, support and protect cell function. Second is to defend against pathogen. Third is to provide shape of the plant cell. The main components are phospholipid and protein. Phospholipid contain both hydrophobic tails and hydrophilic head. Hydrophobic arranged in parallel and facing each other, while hydrophilic facing the aqueous environment. Proteins strengthen the structure and viscosity.
What is the functions of the cell membrane?
Regulating the movement of material into and exiting the cell, selective permeability, give shapes, strength, and protection to the cell. What is nucleus?
Nucleus is the genetic material. Is a eukaryotic cell is found mainly in the nucleus, the most important organelle, typically 5 micrometer in diameter. Structures contain nuclear membrane, 2-layer inner and outer layer. Outer layer is continuous and connected with another organelle, the rough endoplasmic reticulum. Chromatin contain DNA and protein. Nucleolus contain RNA, which is synthesized and aggregate. Nuclear pore, 100 micrometer in diameter, allowing RNA and protein to move in and out of the nucleus. Structures regulating cell activities, which are continuously happening by determining which protein molecule should be produced. Involved in heredity functions. Involved in cell divisions, locations at ribosome synthesis, nucleolus. What are the organelles?
First is mitochondria. Exists in all eukaryotic cell. Higher number of mitochondria, higher level of activity, 1 to 10 micrometer long, move freely in cytoplasm with no defined shape. Function, location, energy synthesis and generation in the cell. Site where energy liberated via cell respiration. Cell's powerhouse.
Second one is plastid. Only exists in plant cell. Three type of plastid is chloroplast, chromoplast, and leucoplast. Most important, chloroplast. Site of photosynthesis in green plant and green algae. Size is 0.5 until 2 micrometer wide, 10 micrometer long. Shape is vary. Locations do not stay in the fixed positions. Structures surrounded by outer and inner membrane. The intensive internal membrane are made up from vesicles which are known as thylakoid. The thylakoid vesicles are flattened to form discs which are scattered to form grana, single disc granum.
Largest structure in eukaryotic cell made up from phospholipid bilayer, a system of sac which enclose a membrane known as cisternae, connected to nuclear membrane, Golgi apparatus, and lysosome. Classified based on the presence and the absence of ribosome, smooth endoplasmic reticulum, and rough endoplasmic reticulum. Smooth endoplasmic reticulum, tubular or sac-shaped, smaller than rough endoplasmic reticulum. The functions are involved in synthesis and transportation of phospholipid, fatty acid, and steroid. Detoxification of hazardous chemicals such as pesticide and carcinogen. Rough endoplasmic reticulum, stacked vesicle with ribosome on their surface. Function temporary storage for protein synthesized by ribosome. Transporting protein from one part to the other part in cell by using cisternae space. What is ribosome?
Occur in all cells, especially the actively synthesizing protein. Consists of two subunits, small and large subunit, synthesized by nucleolus. Locations: freely floating in cytoplasm, free ribosome, and attached to rough endoplasmic reticulum, made up of protein and RNA. Functions: protein synthesis. What is Golgi apparatus?
Occurs in all animal and plant cells. Consists of a group of membrane-surrounded sac. Occurs mostly in secreting cell. Function are processing, modifying, and packing new proteins from endoplasmic reticulum into glycoprotein. Secretion of glycoprotein. Formation of new membrane and cell wall. What is lysosome?
Only existing in animal cell, spherical shape, contain digestion enzyme, 40 types, and formed from the Golgi apparatus. Functions: intracellular digestion via phagocytosis, destroying bacteria and foreign substances in cell. Involved in autolysis, lysosome will be dissolved and all the digestion enzyme will destroy the whole cell. For example, tadpole to mature frog. What is vacuole?
Vesicle filled with fluid which is enclosed in single layer membrane or tonoplast. Sub filled with H2O, organic material such as amino acid and glucose, mineral salt, dissolved O2 and CO2. In animal cell, small vacuole or have none of it. In plant cell, large vacuole and increasing in size as the cell become more mature. Function: storage for waste material, cell enlargement. Water-filled sac will further increase the cell size. What is mitosis?
Mitosis is a type of cell divisions which involve the divisions of the nucleus to produce two daughter cells. Daughter cells contain same number and same kind of chromosome as the parent. Mitosis occur in all somatic cells, all body cells except gametes. Somatic cells contain two sets of chromosomes, one set from female parent and another from the male parent. Two sets of chromosome, diploid cells, 2n minus somatic cell. Cell with single set of chromosome, haploid cells, and is gamete. The cell cycle, two major phases, interphase and mitotic phase, M phases. Interphase can be divided into three subphases. G1 phase, first gate. Cells grow by producing protein and cytoplasmic organelle. Cells grow and differentiations occur. S phase, synthesis. Synthesis of DNA occur. DNA replications. G2 phase, to gate. Cells grow by producing protein and cytoplasmic organelle. Cell grow and differentiations occur. In interphase, chromosomes appear as thread-like structures, chromatid. In general, interphase phase where the cells prepares the cell's divisions. M phase. Once interphase complete, M phase begins, in which mitosis and cytokinesis occur. Shortest part of the cell cycle because interphase make up about 90% of the cell cycle.
What are the stages of mitosis?
Mitosis have four stages. It is prophase, metaphase, anaphase, and telophase. In prophase, centrioles move apart to opposite poles. The chromosomes coil up, condense, and shorten. Late prophase, each chromosome appears as two identical chromatids. Sister chromatids attach at centromere. Nuclear membrane breaks down, nucleus disappear, formations of spindle fiber. In metaphase, centromere attach to spindle fiber. Chromosome move to cell's equator. Anaphase, centromere of each sister chromatids divides into two. Each will spread and move to opposite cells. Spindle fiber pull the centromere toward each pole. When chromatids reach the pole, they will become independent chromosomes. Telophase, two sets of chromosomes at each pole. Chromosome decondense thread-like structures. Spindle fiber disappear. Nuclear membrane begin to form around both sets of chromosomes. Nucleolus reappears. Result of mitosis, two daughter nuclei. Each will same chromosome number as parent cell. Cytokinesis usually occurs between late anaphase and end of telophase. Two mechanisms: cell plate formations, plants; cleavage, animals. What is the process of meiosis?
Involves two nuclear division. First is meiosis 1, second is meiosis 2. Each contain four phases. First is prophase 1 and 2. Second is metaphase 1 and 2. Third is anaphase 1 and 2. And fourth is telophase 1 and 2. Meiosis 1, the homologous chromosomes are separated into two haploid daughter cells. Meiosis 2, sister chromatids of each daughter separated, same as mitosis. Interphase, thread-like chromosomes. Chromosome replicates. In prophase 1, centrioles move to opposite poles. Chromosome thicken and condense. Each chromosome becomes visible, consists of two sister chromatids attached at centromere. Homologous chromosomes pair together, which is synapsis. Each pair chromosomes is called a bivalent, four chromatids. In each pair, one maternal chromosomes and another is paternal chromosomes. Chromatids crisscross along their length, which is chiasmata or chiasma. Nuclear fibers and nucleolus disappear. Spindle fiber is formed. In metaphase 1, pair homologous chromosomes line up at the cell's equator. Chromatids of each chromosome are attached to spindle fibers. In anaphase 1, the pair homologous chromosomes separated from one another and move to opposite poles pulled by spindle fiber. The sister chromatids remain attached. In telophase 1, chromosomes reach the pole. Each pole has a haploid chromosome set. Spindle fiber disappear. Nuclear membrane and nucleolus reappears. Cytokinesis occurs. In prophase 2, central duplicates and move to opposite poles. Each chromosome consists of two sister chromatids attached to centromere. Nuclear membrane and nucleolus disappear. Spindle fiber formation. In metaphase 2, chromosomes line up at the cell's equator. In anaphase 2, sister chromatids separate to become independent chromosomes. Spindle fiber pull chromatids to opposite poles. Lastly, in telophase 2, chromatids reach poles and become new chromosomes. Nuclear membrane and nucleolus reappears, thread-like chromosomes. Cytokinesis occurs and 4 haploid cells form. What are the significance of mitosis?
First is produce new cell for growth. Second is repair and replace cells that are dead and damaged. Third is asexual reproductions such as amoeba sp. Fourth is ensure that the offspring cells are genetically identical to the parents. And fifth is preserve diploid number of chromosomes. What are the significance of meiosis?
The significance of meiosis is allow trait inheritance in offspring, progeny or daughter cell. Genes from both parents are present in zygote. Maintain diploid number in each generation. Ensure production of haploid gametes in sexual reproduction. Produce genetic variation among offspring, which is crossing over.